Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the deep dive. You've brought the sources, and we are here to provide the shortcut. Today, we are undertaking a really essential knowledge sprint. We're diving straight into the structural rules of life, specifically from NCERT Class 11, Chapter 2, Biological Classification.
Exactly. This is a foundational deep dive, and it's tailored for learners like you who need to pull out the absolute core concepts. You know, the distinguishing features, the exceptions, the critical terms that really form the backbone for complex exams like the UPSC. Our mission is to distill the, well, often confusing details of the five kingdoms. We'll be focusing especially on Monera, Protista, and Fungi.
Okay, but before we jump into that microscopic world, just a quick but really heartfelt thank you. We're able to keep bringing this focused, time-saving content to you because of the incredible support from our existing members. You fuel this engine, and we really, really appreciate it. If you find value in these deep dives, please just take a moment to hit that like button, share a comment about what you learned, and if you're capable, please consider joining our paid membership on YouTube. It really helps support this work.
Okay, let's do it. Let's unpack this journey into classification. And we have to start with the most basic question. What defines a living organism? The source gives us a whole list.
Right. When we think of life, we list things like growth, reproduction, the ability to sense the environment, respond to it, metabolism, and the ability to, self-replicate and self-organize. But for an exam, you need to know which of those are defining traits, the ones that apply universally with no exceptions.
That is the critical distinction. Growth, for instance, is a bit problematic. Because non-living things, like, say, a sand dune or a mountain, they can grow by accumulating material on the outside.
And reproduction isn't perfect either, is it?
Not at all. You have infertile couples, worker honeybees, mules, all are clearly alive, but they can't reproduce, so it fails the test.
So if those two major ones fail the universal test, what's left? What survives as the mandatory rule for life?
The two mandatory, universally defining characteristics of life are metabolism and cellular organization.
Okay, metabolism and cellular organization.
Yes. Metabolism is every single chemical reaction happening inside a living system. There is no known non-living object that shows metabolism, period. And cellular organization is the foundation. Life just can't exist without that basic structural unit of the cell.
Just to cap off that idea of complexity in living things, the source notes the most evolved feature we know of, self-consciousness, the ability to be aware of oneself.
Which is, generally accepted as unique to humans. It's the top of the pyramid, so to speak. So now that we've defined life, we can turn to how we organize it. How do we manage the millions of species?
That's taxonomy, right? The whole process of identifying, naming, and then classifying everything.
Exactly, taxonomy. And it leads directly to this hierarchical system.
It's like biology's librarian, always trying to put things on the right shelf. We use categories or taxa at different levels. How does that hierarchy actually flow?
We start with the most specific, which is the species. Then we move up to genus, then family, order, class, phylum, or division, if we're talking about plants, and finally, kingdom.
So terms like wheat or mammals or fungi, those are all taxa, just at different ranks in the system.
Precisely. And here's the crucial insight, the thing you need to remember as you go up that ladder.
What happens to the common characteristics?
As you move higher up the taxonomic hierarchy, so from a specific species up towards the vastness of a kingdom, the number of common characteristics shared between the members, it goes on decreasing.
Ah, okay. So members of a species are almost identical, but members of a whole kingdom, like Animalia, share way fewer basic traits.
Exactly. It's the conceptual price you pay for grouping things more broadly. It's a fundamental rule of this system.
That makes perfect sense. So now let's get to the big framework itself. R.H. Whittaker's 1969 proposal, the five-kingdom classification.
Yep. We're talking Monera, Protista, Fungi, Plantae, and Animalia.
And this was a huge change, wasn't it? It was a massive correction over what came before.
Oh, absolutely revolutionary. It recognized that just having a cell wall wasn't enough to just lump an organism into Plantae, which was the old way of doing things.
Wait, so just to clarify, why were fungi, Monera, and Protista originally stuck in with the plants? And what was the big reason Whittaker's system kicked them out?
Well, the primary reasons were cellular structure and nutrition. Fungi, for instance, were removed because their mode of nutrition is completely different. They're heterotrophic. They don't make their own food. Plants are autotrophs.
And Monera.
Monera are prokaryotic. They lack a true nucleus. Everything in Plantae is eukaryotic. Plus, the very structure of their cell walls was fundamentally different. You just couldn't keep them grouped together logically.
That distinction, prokaryotic versus eukaryotic, autotroph versus heterotroph, that is the key breakthrough. Let's dive into that first kingdom then, Monera, the bacteria, the ultimate prokaryotes.
Indeed. They're the oldest and most adaptable life forms. You find them, well, almost everywhere, even in places hostile to everything else, hot springs, deep oceans, as parasites. And we first classify them by their basic shapes. You have the spherical coccus,
The rod-shaped bacillus.
the comma-shaped vibrio, and the spiral spirillum.
Okay, and within Monera, we split them into two major groups, Archaebacteria and Eubacteria. Let's start with the oddballs, the Archaebacteria, the ancient ones.
These are the extremophiles, the salt lovers, the heat lovers, and a really key example, the methanogens. They survive in these brutal environments because their cell wall structure is unique. Crucially, they lack the peptidoglycan found in most true bacteria.
And that structural difference is what gives them that extreme resistance.
Exactly. And the methanogens have a really important role in ecology and even in, applied science.
Right, because they're in the guts of animals like cows and buffaloes.
Absolutely. They're obligate anaerobes. They live in the guts of ruminants, they break down cellulose, and they release methane. That's biogas.
So there's a direct link from microbial classification all the way to renewable energy.
It's a perfect example. Now, for the Eubacteria, the true bacteria, they have that rigid cell wall, a flagellum if they're motile. And this is where we find a group that's vital for our discussion, especially for previous exam questions.
You're talking about cyanobacteria.
Yes. Let's zoom in on cyanobacteria, what people used to call blue-green algae. They're photosynthetic autotrophs. They have chlorophyll, just like green plants.
Which is why they were misclassified for so long.
Exactly. But the real gem, the PYQ connection, is their function. They fix atmospheric nitrogen.
And that ties directly into the concept of biofertilizers, which is a common topic. How do they do it? What's the mechanism?
They do it in these specialized, thick-walled cells called heterocysts. Organisms like Nostoc and Anabaena have these structures, and they convert inert atmospheric nitrogen into usable compounds for plants.
So that ability to enrich soil naturally is precisely why Nostoc shows up in the sources as an effective biofertilizer. That's a critical application to know.
It is, and that neatly transitions us to the next kingdom, Protista. If Monera are the single-celled prokaryotes,
The Protista are the first single-celled eukaryotes.
they're the bridge kingdom. They have a well-defined nucleus, membrane-bound organelles. They can reproduce asexually and sexually. A lot of complexity there. They're primarily aquatic, and we need to look at a few of their key subgroups.
Okay, let's start with the Chrysophytes, the diatoms and golden algae.
Diatoms are fascinating. Microscopic plankton, mostly photosynthetic. Their cell walls are unique. They form two thin, overlapping shells that fit together like a little soapbox.
And when they die, that's what creates diatomaceous earth.
Exactly. When diatoms die, their silica cell walls accumulate over billions of years.
Which is that powdery stuff used in polishing, filtering oils and syrups. Another huge real-world application from a tiny organism.
It is. Now, there are also the Dinoflagellates. They're known for their two flagella and their varied colors. But the most famous example is when they have a massive population explosion.
The red tide.
The red tides, yes. And they can release toxins that are powerful enough to kill marine life. A huge ecological impact.
From a unicellular eukaryote, we also have to mention the group that's had just a staggering effect on human history, the sporozoans.
The sporozoans are identified by having an infectious spore-like stage in their life cycle. And the key example, which the source material notes for its global impact on human population, is Plasmodium.
The malarial parasite.
The malarial parasite. It's a stark reminder that these classifications map directly onto major human health crises.
Okay, finally, let's turn to kingdom fungi, the ultimate heterotrophs. We see them everywhere on moist bread, rotten fruit, and of course as edible mushrooms like Agaricus.
Their uniqueness is all about their lifestyle and structure. They are heterotrophic, they can't make their own food, they can be saprophytes, parasites, or symbionts. Their body is made of these long, slender, thread-like structures called hyphae, which form a network called the mycelium.
Their sexual reproduction process sounds way more complex than Monera or Protista. There's that intermediate stage we need to focus on.
That is the most complex detail here. Sexual reproduction has 3 steps. First, plasmogamy, the fusion of protoplasts.
Okay.
Second, karyogamy, the fusion of the 2 nuclei. But, and this is the key, in many higher fungi, karyogamy is delayed. It doesn't happen right after plasmogamy.
So what happens in that gap?
In that intermediate stage, the cells contain 2 nuclei, one from each parent. This condition is called a dikaryon.
A Dikaryon. Okay.
And that phase of the life cycle is called the dikaryophase. It's only later, often inside what we see as the fruiting body, that the parental nuclei finally fuse, become diploid, and then immediately undergo meiosis to produce haploid spores. Understanding that specific phase, the dikaryophase, is essential.
And their classification into 4 classes is based on the mycelium, spore formation, and those fruiting bodies. Can you give us just a quick rundown?
Sure. You have the Phycomycetes, like mucor, then the Ascomycetes, or sac fungi, which includes things like penicillium and yeast, then the Basidiomycetes, the club fungi, which is where you find your common mushroom, Agaricus, plus rusts and smuts.
And lastly, that confusing group, Deuteromycetes. Why are they called imperfect fungi?
It's kind of a holding group, a wastebasket. They're called imperfect because we only know their asexual or vegetative phases. The moment scientists discover the sexual stage of one of these fungi, it gets moved immediately to either Ascomycetes or Basidiomycetes. They're only imperfect in terms of our knowledge.
Which brings us full circle to that application question we talked about. Which of these Agaricus, Nostoc, and Spirogyra are used as biofertilizers?
And our deep dive gives the clear answer. We confirm that Nostoc, which is a cyanobacterium from kingdom Monera, has heterocysts and fixes nitrogen. That makes it a powerful biofertilizer.
Whereas Agaricus, the mushroom, is a fungus. It doesn't fix nitrogen.
Not at all. And Spirogyra is an alga. So connecting that specialized cell in Monera to a global agricultural practice, that's the payoff of studying this level of detail.
So what does this all mean for you, the learner? We've covered the crucial features of life, the framework of taxonomy, and the unique testable features of Monera, Protista, and fungi.
Just remember these nuggets. 1. Define life by metabolism and cellular organization. 2. Remember that shared characteristics decrease as you go up the hierarchy. 3. Monera's utility is in the cell wall of Archaebacteria and the end-fixing heterocysts of Cyanobacteria. And 4. Protista gives us diatomaceous earth and the parasite Plasmodium.
A really concise, detailed breakdown.
Now, here's a final provocative thought for you to take away. Whittaker's system completely revised Plantae based on observable differences, right? Cell walls, nutrition.
Right.
But today, with rapid genetic sequencing, with ribosomal RNA analysis, scientists are constantly finding genetic relationships that completely overturn what we see with our eyes. So my question is this. What other stable multicellular kingdoms, maybe even Animalia, the one we think we know so well, might be due for a dramatic, revolutionary reclassification based on genomic evidence we're only now starting to fully understand?
That is definitely something to ponder as you keep exploring the limits of biology. Thank you for joining us for this essential deep dive.
And remember, if this detailed conversation was useful for your studies, please support us. Take a second to like this video, share a valuable comment, and if you can, consider joining our paid membership on YouTube. A special shout out again to all our existing members. Your contributions are what allow us to create this kind of detailed, time-saving content.
We'll see you next time on The Deep Dive.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.